So this demonstration video 3 is a brief introduction to differential expression analysis in practical terms using limma, which applies a linear model. So that linear model tries to take into consideration the experimental conditions that are of interest and how that relates to the biological response of the different reads and isoforms of genes that are of interest. We can describe the condition, the effect of the conditions of interest, things like the foot-mouth infection status and the read on the response of the reads. And if you consider other sources of variation as well. So what we're dealing with in the more practical terms, when we're using limma, the bioconductor package, is that we're typically looking at a expression set, which is a matrix-like data set, which we obtain from the Callisto H5 bootstrapping data that contains quantitative values for each feature for each transcript across many samples. It also stores other metadata, explaining the context and so on of different experiments that are applied. In general, what we're doing here is this particular response of the reads that are found at a particular transcript, and to see whether the treatment here listed as a beta coefficient by X, some treatment, plus the intrinsic variability associated with each particular sample, to see if the beta is large or maybe it's actually close to zero. As mentioned in the introductory videos, the variances could possibly across similar features that are similar values to get a better estimate of the pooled variance. That shrinkage means that we get more accurate models that are of interest. So we can consider that we have several steps here to apply in practical terms. So we have to set up a design matrix to compare what groups are we comparing. We want to define the comparison that that's of interest, fit a linear model, fit a contrast model, contrast where possible, empirical Bayes moderation of the features can mean that we can generate then the key metrics such as the F value, log to fold change, adjusted P value, and so on to assess further. In limma, the simplest option for the design matrix does not need a formal contrast. So here we might have some X value encoded as either 0 or 1, depending on two groups, where you have YG is equal to either beta 0, which is kind of an error, plus beta 1 by X. So therefore, as X1 is 0, we get two different groups, and therefore beta 1 denotes our magnitude of change that we observe. The other option is a contrast where you have more complex design with multiple factors. So for any gene G with the first factor set to 1 for group A and 0 for group B, we can compare those two groups. And we might have other factors like X1, sorry, that's X0 first, X1, then is X1 equals 0 for A and X1 equals 1 for B. We have YG is equal to a function of beta 0 by X1 and beta 1 by X1. So if we apply this to our models that are of interest, we can think of it in this kind of more simplified terms that we might have a breed where we're classifying them to two different breeds for simplicity here, although we have obviously have three, and we have foot-mouth status as either present or absent. So it's present, so X0 equals 1 for present foot-mouth infection, X0 equals 0 for negative, and X0 equals 0 for MG and 1 for non-MG. Thus, for the first class where we have FMD infected MG cattle, YG is product of beta 0, the infection status plus other errors, other factors not accounted for in our model. For the non-MG, it's the same as above, but with beta 1 added. Thus, we can compare and contrast the first model versus the second model to estimate beta 1 and the interaction between beta 0 and beta 1. Then we have the FMG negative and MG, so they have no other factors. They're kind of our baseline, so you can compare them to get a better estimate of beta 0 for our first model. And then we have those that are FMD negative and non-MG. Thus, we can estimate beta 1 more effectively and estimate this model more effectively as well. So that's kind of our basic setup. And now we'll move on to looking at importing our libraries into Bioconductor and using TX imports. So we'll have to install that using BioC Manager next. And we'll do that now on the server. So now we're on the server and we can use that RMD file to firstly check that we've loaded the the correct tool that we want for our library here. So we're using TXI import. We install that with file conductor. So you would use the command here that we just commented out here to install that if we needed to. What we're going to do is use our file path from the full data frame. So you see here, if we go full data path, we have the file paths that we need. So we need the file paths to know where we're going to get this data. So hence we have this expression here where we have the abundance.h5 file at the end, or paste that onto the existing path, separated by the / so the files correspond to the files that are of interest here, numbers 1 to 15. And then we can use tx import to import these as Callisto type files, where we're sending this to the txi Callisto variable. So you can see that's being read in now. And if we look at the head of that file, we'll see, or indeed, a lot of information about the different features that are present. So what we can do next, while that's printing to screen, is activate limma. Again, that's installed via bioconductor, and we're also going to use the deg list function from edgeR, so that we can import the count data from Callisto. So if we look at the structure of Y here, we can see that we have a lot of information. We have 38,000 transcripts that are being expressed across the 15 samples that are of interest. So what we can use is we can set up the design that we're having here. So where our design is that we have a breed, a condition that's infected or uninfected, and then we have the Holstein Friesian set, and then the Malinois Geta, and then the others are the Halicar breed that aren't shown here. That's our initial design. We could also have a simpler design, for example, if we wished, where we just looked at the condition only. So let's try that as the design. So we have a more simple setup here. So we're going to keep the ones that have reasonable amounts of expression, and take a subset of Y that with those values only. And we see there we have, in this case, 19,000 genes, because we're just looking at the condition only. We then normalize this using the calc norm factors function, and send that to the zoom function to adjust Y for our design of interest. So we see that we have our data all together there. And what we're going to do next is fit the linear model with that's in limma based on our input V and our design. We're then going to use the empirical base to extract key information about that, including the estimates of key metrics that we need. And what we're also going to do is use our FitM here. So FitM has a range of different values. Arbitrarily, we're just using the coefficients. So if I FitM, if I spell it properly, coefficients. We can see that if we get the numbers of them, this contains the number of isoforms that we have present. Hence, we can put this into the top table function to extract the table of all our genes into out1. So if we look at out1, then we just look at the top of it, we can see there that we have the log2 fold change, the average expression, the t-value, the p-value, the q-value, and the beta value as well. So that means we can use the ggRepel package, which we've installed via CRAN, then to plot this information as well. So that is a useful aspect to have. So we can initially create a path where we're reading in out1, the out1 data. We're taking not taking the f value and x, we're taking the log fold change. And then we're taking the adjusted p-value, and we're taking the log 10 of that. We're setting the row names to be those of out1. Sorry, that's not very visible. Let's start with that only, because we're going to add labels here later for GG repel. But let's just start with this simple function. So that creates a basic background for a volcano plot. And then we can add geom point on top of that just to create some points. And it's best to just build this up very simply first. So we see our points there. So you see this doesn't maybe quite look like a typical plot, but this is because most of our adjusted p-values are actually are the minus log base 10 scaling of them has set them all to 0. So what we can do is scale that differently so they have a adjusted size and an alpha for those plots there. We can see we can add on information onto the axes labels as well to make that more informative in terms of the log2 full change on the x-axis and then the log 10 of the false discovery rate on the y-axis, and then we can add horizontal lines on the x and y axis to note ones that might be of interest. So if you see there's no substantial changes in expression here, which is unsurprising because we have a very small sample size, so we're not necessarily going to pick them up if there were any present. If we do a more advanced model, we can reapply that using the condition plus the breed. And that's kind of the same stuff we will here we'll get 21,000. We didn't get 21,000, we got 19,000 samples. So that's using design where we incorporate the breed as well as the condition. We use the keep function to take a subset of isoforms for y. So we have 19,000 there for y. We again normalize with the calc-norm factors function, and then we can look at the structure of V. So we see we have our data in that form. Then we can apply linear model fit again based on the design, just as we did before. And we can even use the plot SA function to plot the change in expression as we have higher expressions. That's looking at the variance in expression on the y-axis. So you can see that it increases slightly as we move along, although it might be distorted by certain outliers. You see for the 0 values, the minimum values certainly have higher variation in expression. So what we can see then is that we have a new out one table, and we can look at those values. Bear in mind that because we have multiple conditions here, we have the condition of the unaffected ones, the Holstein Friesian and the Malna Gita, and the average expression overall, and the F value corresponding with that complex function. So what we can do is even then look at the design simply based on breed in the same way as before. So we're just setting it to breed only. So now we have about 19,000 isoforms of interest as again. And again, we can create our table out one. If we look at out one, we can see there we have the Holstein Friesian breed, Malna Gita breed, the F value associated with that. And we can also look at the fit M, which should be the same as what we had before. So you can see there that we're able to test a range of different things in this single data set, and the best model obviously incorporates the multiple variables together. And you'll note that volcano plots here don't look the same as what we might expect with other data sets because we have quantified the p-values in a slightly different way for this kind of likelihood ratio test, so we don't get the same distribution that we might otherwise get. So if you have any questions on this, please ask me during the labs themselves.